This presentation is titled Prompt Engineering, Gene Prediction in GeneMarkS and GeneMarkS-2. First, to begin with a presentation overview, we'll be describing some unsupervised gene prediction pipelines, which are GeneMarkS and GeneMarkS-2. And as we'll describe in the workflow and different diagrams, which can help demonstrate to us how different data is processed and how parameters for these models are set, we're first going to have to provide input FASTA files, which help determine base parameters that the model uses, from which it attempts to make predictions of other genetic sequences from the parameters which are set from an unsupervised training model. And we'll be able to characterize the output of two different pipelines, namely GeneMarkS and GeneMarkS-2, under different FASTA input files. And we're going to discuss and think about whether any sequences which are produced in GeneMarkS-2 from the input FASTA files, which either contained a little more than 5,000 sequences, around 3,000 sequences, and for the third choice, around 1,800 sequences, and finally around 800 input sequences, determine the total number of sequences that the model can produce from the output, and how informative this can be for us when we're trying to predict novel types of genes and other types of characteristics of genetic data, or in genomics in general. So the reference that we're going to be looking at is GeneMarkS. It's a reference which is describing an unsupervised training method that these authors from Georgia Tech looked at. And recently, maybe like around 20 years later, they further iterated on this GeneMarkS procedure by making another pipeline, GeneMarkS-2, which they also made publicly available, and which can serve a similar use for predicting microbial genomes or genes in microbial genomes, as well as genes in prokaryotic genomes, which is of interest for us. And just very briefly to describe the protocol workflow, I described this in another video that I have on my channel, in which I described how one can make use of GeneMarkS through the online resource that they have to determine or to predict different types of genes. And this algorithm for GeneMarkS-2 very much relies on the same type of workflow, in which there are three initial steps, as well as the main cycle, which is given by steps 4 and 5 in the bottom half of the protocol workflow. And all of the initial steps help us determine how the different parameters are set for the model from some input genomic DNA that I'll also describe we can pass to the algorithm with FASTA files. And by looking at how the change in the number of sequences of the FASTA files is different amongst different input genomic DNA files that we put into GeneMarkS-2, how that would impact the total number of sequences that the algorithm could predict from its unsupervised training procedure. So this is just a general overview of the 5 steps that are involved in this algorithm for it to be able to make its predictions. And when we look at GeneMarkS and GeneMarkS-2, the output of both of these unsupervised pipelines alike is dependent upon FNN output and FAA output. These are just two different types of file types which are used to process targets that are given from the FASTA input file. And then this is helpful for us to make to obtain the full length sequences afterwards that we would like to gain further information on to determine what's the length of the introns, what's the length of the exons which make up the gene, as well as any type of potential conserved sequences which are amongst these genes. And for the FAA output, we can have a similar type of sequence with the only change is that the FAA output is given in the amino acid sequence format, and in comparison to the DNA output, which is given from the ATCG base pairs. And we can see that from the first input FASTA file that we feed to GeneMarkS-2, that there are 5,761 sequences produced. So what we're going to try to see in line with many modern with many modern interests in machine learning, is determining how we can prompt and make different use of this AI machine learning pipeline for determining which set of genes that are predicted from the pipeline given a certain training data set are the most informative for our purposes of gene discovery and gene prediction. And in the second case, when we feed a different type of output FASTA file, which is given by a FASTA file with approximately half of the total number of sequences of that we were providing in the first FASTA file, the GeneMarkS-2, we can see that there are around 3,100 sequences. And in comparison to the previous sequence that was given in the last item from sequence 5,761 from the first input FASTA file, this genetic sequence is a little bit shorter. And we can also see from the corresponding FAA output that this sequence is even shorter as well in comparison to the last sequence that was given in the FAA output from the first FASTA training file that we provided. So already we're able to gather and glean that depending upon how many model parameters we can instantiate for GeneMarkS-2, the gene prediction quality, it can proportionately suffer in being able to predict approximately half as many genes, depending upon how much we cut or how many sequences we remove from the FASTA file, which we first use as the DNA input for the pipeline in the very first step before the 3 initial steps are processed. And then we can obviously conclude that or anticipate that similar observations apply for the remaining FASTA files, in which for the third FASTA file, there are around 1,700 sequences produced. And in the fourth FASTA file, in comparison to the 3 previous FASTA files, besides only having around 700 sequences produced from the model from GeneMarkS-2, the unsupervised training procedure is actually able to suffer, or it suffers a lot more in that the sequences and the total length and complexity of the sequences that are produced from the third and fourth FASTA file also continue to be a lot less informative and can be less indicative of how different types of genetic elements in the sequences can interact with one another. And so the conclusions that we have is that in addition to some previous experiments and different types of ways that we can make use of gene prediction pipelines that I described in a previous video with GeneMarkS, we can also see that for GeneMarkS-2, we can observe that from another unsupervised gene prediction pipeline, we can engage in prompt engineering, which is just basically trying to look at how the output of this AI model or the machine learning pipeline would differ depending upon the number of input sequences and hence the number of parameters that we can observe. And recall that from the total collection of the FASTA files that we have been giving and describing, that from each one of the 4 FASTA files, if we approximately delete by 50% fold reduction in the number of input sequences in every one of the FASTA files, that the output of GeneMarkS-2, it not only suffers in that it's not able to make as informed predictions for more complicated genetic sequences, but also that the gene sequences themselves are much shorter, which is related to the intermediate output of a hidden Markov model. And then we demonstrated how these themes for prompt engineering and determining how the output of different AI and machine learning pipelines would fare is dependent upon the different output formats from the algorithms. And in this case, the output formats were FNN and FAA. And thanks for watching.